Hi everyone, and if you've made it to this video, you have officially made it into Module 2 of Unit 1 Biology. And our first topic is going to be on nucleic acids, namely DNA and RNA. Now, this is a brand new topic. You would not have done this at the CSEC or Form 5 level. So we'll take a little bit of time to go through everything. So I'll make it into a two-part video so that it doesn't become overly long. Alright, so in the first video, which is today, we will be taking a look at mainly the structure of DNA, and also we'll be looking at some of the functions of DNA and replication as well. Okay, so all nucleic acids has the similar structure, meaning it consists of a base, a sugar, and a phosphate molecule. Those three things make up a nucleic acid, or rather a part of a nucleic acid, right? It's actually called a nucleotide. You could think about this as like the a single part of a nucleic acid. A nucleic acids are made up of many nucleotides, right? So this is a simple drawing of a nucleotide, and I color coded it here for you so you could see the different parts of it, right? So in blue we have the base, right? So the bases, there are 4 different bases in a DNA molecule, but there are 5 bases overall shared between DNA and RNA, and we'll get to that when we do RNA, right? But for now, we can look at 4 different bases, right? So 2 of them are called purines, which is adenine and guanine, and 2 others are called pyrimidines, which are cytosine and thymine. So any one of these 4 could make up a nucleotide. Right, so the little molecule in blue there, you don't need to know how to draw exactly the base, right? Just know that that's how it looks, right? Like a hexagon, right? In purple, we have the sugar. So in DNA, the sugar is called deoxyribose. Okay, that's what gives DNA D, right? Deoxyribose. The final part is the phosphate molecule, and that is in green, and it is bonded to the sugar via 2 places, either on the 5th carbon or the 3rd carbon, and you can see that circled in red there. If you remember from the carbohydrate video, we like to count the carbon in a certain order around the molecule, and that is very important here because you can see that the phosphate is bound to any one of these 2. Okay, and that is actually very important. 3 and 5, just keep that in mind when we do the replication later on. Okay, but this is the general structure of a nucleotide. So let's take a little bit of notes here now. So a nucleic acid is a large molecule, and DNA and RNA are prime examples of them, and they are found in living organisms, and they control essentially the characteristics of an organism by ordering how proteins are being made. They are essentially brains of all the cells of the body. They are found in the nucleus. Okay, so we look at DNA today, really. So DNA is a double-stranded molecule. Now, everyone would have seen how a DNA molecule looks like, whether you watch movies or you actually read about it. You know how it looks, right? So it's actually made up of 2 complementary antiparallel polynucleotide chains, and they are held together by none other than hydrogen bonds, right? So we'll take a look at what each of these words actually mean, right? Complementary and anti-parallel. All right, but first let me just take you through the structure one more time. All right, this is a more professional drawing of the a nucleotide, right? So each nucleic acid is built up of a number of nucleotides, and each nucleotide is built up from a nucleoside plus a phosphoric acid. So these 2 molecules here are called the nucleoside, and the nucleoside is bounded to the phosphate group to make the nucleotide. All right, so the nucleoside then is simply the sugar and the base. All right, and as I said, the base could either be a purine or pyrimidine, and there are 2 of each that you will have to learn, right? Very, very important. Okay, And we know that DNA contains the sugar deoxyribose, right? That is essentially one of the main differentials between DNA and RNA. All right. So the structure of a DNA molecule is a double-stranded helix. Now we did these in past videos, right? So you could always go back through those carbohydrate videos to get a look at what a helix looks like, but you'll see it in this video as well. Okay. So it's very important that we make note of the 3' and the 5' end, right? So at the 5' end, the phosphate group is found here, right? That's why we name it the 5' end, because the phosphate group is actually bounded to the carbon 5 of the sugar, right? And at the 3' end, the carbon 3, the third carbon of the sugar is bound to the phosphate group, okay? And then we have two chains coming side by side to one another, and they will be bounded to each other via the bases, which I will show you in the next page. All right. So here we have in this diagram the 4 bases here, right? I will let me just circle it here for you. So purines could be adenine or guanine, and pyrimidines could be cytosine or thymine. And this is the uracil is only found in RNA, so we will be ignoring uracil for the purposes of this video, right? So for now, we take a look at adenine and guanine, and cytosine and thymine. All right. So this diagram here is simply 1/2 of the chain, right? So you could see that they kind of took the time to draw out each nucleotide there, right? You have your sugar here, you have the base here, and you have the phosphate there in yellow, right? So this is called the 5' end, because if you count the carbons, 1, 2, 3, 4, and there's the 5th one here, you can see the phosphate is bound to the 5th carbon atom. That's why we call it the 5' end. And this one down here, and this end will be called the 3' end, right? Because there's a bond at the 3rd carbon atom, right? So now let me show you how the chains pair up, right? So we call it antiparallel because one chain goes in the 5' to 3' direction, meaning you could see that the 5' end is on top over here, and the 3' end will be down here, and then the complementary chain will be in the 3' to 5' direction, where the 3' end will be on top and the 5' end will be on the bottom. So that's why we call it antiparallel. Now it's called complementary now because only certain bases could pair up. All right, so it's the bases that kind of attach the two chains together to make the double helix, right? So we could see that thymine will only bind to adenine. Cytosine will only bind to guanine. All right, so you can see again adenine, thymine, guanine, cytosine. There will never be a adenine guanine. Okay, there will never be a cytosine thymine. The bonds could only be thymine to adenine, or adenine to thymine, or cytosine to guanine, or guanine to cytosine. That is how the bases pair up, right? And they will never be any different from that. Okay. So you don't have to know how to draw this when you're drawing it, right? This is just a representation to show you why they are complementary and anti-parallel. All right. So we could actually predict what our complementary chain is going to be. So if I give you a little exercise and I say, well, the bases on the first chain is going to be A, C, T, and G, what would the complementary bases be on the complementary chain? Well, A will bind to a T, C will bind to a G, the T is going to bind to an A, and the G will bind to a C. So that will be the bases that would pair up the two chains there, right? So it's quite easy once you remember that they only pair in that fashion, right? So thymine to adenine, cytosine to guanine. That is the most important part of this diagram here. Okay. Now let's move on to the first big subtopic of DNA, which is called DNA replication. So you would have done mitosis and meiosis in Form 5, right? And you know all these steps and stuff, right? But let's take a look at the first step, which is interphase. All right. So in interphase, you know that the cells are preparing to divide. All right. So in this phase, each DNA molecule is actually replicated exactly. All right. So an identical copy will be made. So to do this is quite a long process. It's very complicated actually. And 4 enzymes are required in total to do this, right? So DNA helicase, primase, DNA polymerase, and finally DNA ligase in that order. All right. So I have this this diagram here, right, which is I will explain it in full detail for you, right? So you will not be confused, right? So in the first instance, the DNA, which is originally double-stranded, so you could see double-stranded here, right? The DNA has to unwind, right? So it will be unwound or unzipped or whatever word you decide to use, right? So you're kind of unzipping, you could see like a zipper, right? The double-stranded molecule, and it will unzip into single strands, right? So the enzyme that does this is our friend DNA helicase, right? So if I take you back now to the paragraph here, the double-stranded DNA molecule is unwound, right? And it is done so by DNA helicase. So the chain is going to separate into 2 separate chains, right? And each of those will act as a template for the new chains to be formed, okay? And we like to call this semi-conservative replication, which I will go into more detail as soon as we finish this, right? So if you look carefully, the gray chain is the original DNA molecule, right? In this diagram, they kind of did everything in one shot, right? But the gray chain is the original DNA. So we could see the gray chains here, right? So if you pretend that the red part of it does not exist, you could see that the gray part is the original part, and over here in the bottom here has the gray part as well, right? So we could see that it is one strand that it unzips into. Okay, let me draw that over for you here, right? So initially, this is a simplified version here, right? So initially we have the DNA, right? Forget about the winding and whatnot, right? So now helicase is going to come and start unzipping. When that happens, it will now look like this. So you have the last part of it here, but now it's going to unzip like that, right? So that is how it looks after DNA helicase is done with it, right? So the next thing now is primase. So what primase does, it synthesizes RNA. Now we didn't do RNA yet, and we will do it in the next video, right? So RNA is necessary because the main guy in replication, which is DNA polymerase, which is the third enzyme, it cannot work on its own, right? So it needs a little help to start off, right? It can't start a whole chain on its own, but once it has a little bit to start with, it will mash up the place and just replicate the entire chain, right? So primase synthesizes, as you can see in this diagram, a very short segment of RNA, and we call it the RNA primer. You know, primer is the first thing. Primer means to start, right? So the primase will do that, and then DNA polymerase will take over. So this goes here, right? So you can see the red is the new DNA, right? So from here, DNA polymerase will take over and start synthesizing an exact copy of the strand, right? In the same antiparallel fashion, right? Complementary fashion, where the bases will be complementary to each other. It's the same thing as we did in the first page, right? So that is in the 3' to 5' end, right? So as you can see, when the chain unwinds, one of the chains is going to be the 3' to 5' end. All right, that's 3 to 5 here. And then the one on the bottom is going to be the 5' 3' end, right? Now, for some reason, DNA polymerase works very well in the 3' to 5' range, right? It would only go forward from the 3' to 5'. It does not work that well for the bottom piece, which is the 5' to 3'. It doesn't know how to work. It could only go forward, right? It can't work backward. So this is where DNA ligase comes in. So DNA polymerase could still try and synthesize chains on the bottom strand, right? But it could only do so in little fragments, and we call it Okazaki fragments. I don't know why we call it that, but it's cool, right? You could remember it because it's cool, right? So we have little Okazaki fragments there, but they are fragments, which means that they have little gaps in them, right? So what DNA ligase does, it joins the fragments together to make a smooth chain, right? And this is essentially what DNA replication is, right? So there are 4 main steps. DNA helicase unzips the original DNA molecule into 2 separate chains. Then you have primase coming to prime the 2 chains. DNA polymerase comes to add on the bases and add on the chains, the complementary chains to each chain. And finally, DNA ligase will come and join the Okazaki fragments at the 5' to 3' end. Okay? And obviously your diagram, when you are drawing it, does not need to be so detailed like that. I actually drew it here much simpler for you here, showing the steps, right? Obviously, you don't want to make it look that untidy. You know, you could take some pride in your work, right? But this here is what I mean by how DNA polymerase works, right? So let's say, let me see if I could erase the little parts here, right? So, right. So this was the original DNA molecule, right? This was some of the bases on the DNA molecule. And we know A goes to T, C goes to G, T goes to A, G goes to C. Those things, right? We know the bases by now, how they pair up, right? Right. So now, when the chain split apart, you will have one chain, which is this one. That's the top chain, right? And then this chain is the bottom chain. Okay, so what DNA polymerase does now is add on bases to these chains here. So therefore, DNA polymerase will add on a T to the A, G to the C, A to that T, a G to that C, and a C to that G. And this now will continue on through and make its own DNA molecule. Right? Isn't that the same molecule as what we started off with? Right? Look at the big A and the big T. It's the same, is the exact copy, right? So that's how it replicates. And the same thing will happen for the bottom chain. The T will bind to the A, the G will bind to a C, the A will bind to a T, the G will bind to a C, the C will bind to a G. And this will make the bottom strand, and that will eventually finish out. And now you could see that we have 2 exact replicas of the original DNA molecule. Okay? So that's how DNA polymerase adds bases on and creates a new chain, right? Now, the last thing I want to take a look at is this term here, semi-conservative replication. And to understand this, we will simply look at an experiment called the Michelson and Stoll experiment. So, what happened here is they did an experiment with the replication of bacteria. Right, a certain one called E. coli. That's a pretty common bacteria, right? So the E. coli was grown in a lab, right, using isotopes of nitrogen. Isotopes meaning, have a different neutron number, right? But that's not important right now. So some E. coli was grown in this nitrogen here, which is 15, right? The atomic number 15. And you could see the color coded it here in a dark blue. The E. coli now, after you let them grow a little bit, you transfer them over now to a different source of nitrogen, nitrogen 14. And the color coded this light blue, right? So the original DNA of the E. coli would have been dark blue, right? You could just consider it as dark blue. So let me just draw something here. Right. So you could consider the DNA, double-stranded, helical, right, of the initial E. coli as dark blue. But now, when you put them into the light blue solution, and they start to replicate with this new source now, they're going to change. The DNA going to change a little bit, right? So you don't need to understand really the different densities of the DNA, right? That's just because the food source is essentially lighter. This one is lighter and this one is heavier. So that's where the density comes in, right? So what they noticed now was that when you put them into the light blue solution, one strand will remain the same, but the other strand now, it changes, right? It changes color. It changes into the light blue. But why is that, right? Why isn't the entire thing light blue now? Well, this is where the whole thing about semi-conservative replication comes in. It's only half conservative. Conservative meaning that, you preserve the whole thing, but we say this is semi-conservative, meaning there's always a part of the original DNA molecule in the replication, right? No matter what, right? So let's take a look at what happened here. So this is the parent, the first bacteria that was grown in the N15, right? Now, when you put them into the N14, remember the DNA is going to split into two. So this initial dark blue one here would have been split like this. So you will have one so, and then you would have one like this. But now every new replication is going to have the light blue. I do have light blue here, so I'm just going to use green, right? So when you split them apart, yes, each strand is going to keep part of the N15, but now the new strands have to use the N14 as the new source, right? So each of them now, after the first replication in the N14, is going to have part of that N14 with them forever, right? But you still end up with some of the N15 one, right? And that's what we mean by semi-conservative, right? So there will always be a part of the original DNA there. However, when they replicate again, you could imagine that, remember, it have a light blue strand here now, so that is going to be on its own, and then it's still in the light blue solution. So that strand now, that is light blue, will just pick up our next light blue, and it's going to become like this. However, the same dark blue strand, it will remain, and it will pick up a light blue. So it's still semi-conservative, you see? You still have a light blue there. A dark blue, sorry. And in the second one will be the same story, right? The dark blue still persists. And this is what we mean by semi-conservative. So as I said, DNA is a new topic. It's going to be a little bit long, so I will end the video here today, okay? And we will finish it in the next one.